Bismillahirrahmanirrahim. We start A-level biology, and this is 9700 paper 5. This is the first paper 5 that I'm uploading for the students who are taking paper 5, and this is a solved one in which you will learn the techniques how to do a paper 5. And this is the November 2019 paper 5 2, and I hope I can finish it in this half an hour, but otherwise then we'll go into the part 2 of this paper. Now the important thing to understand is that paper 2 and paper 5 are both on the same day. But look at the time and look at the marks. They are both for 1 hour 15 minutes. They are both for 1 hour 15 minutes. But paper 2 is for 60 marks and paper 5 is for 30 marks. So that is a very important thing for you all to understand, that you can't say, okay, well, I finished the paper in half an hour and I came out. Well, that's not the case. Why is it for 1 hour 15 minutes is because it is a lot of paper in which you need to think, you need to read, you need to conclude, you need to do a little bit of homework, you need to do a little bit of written work on top of it. I mean rough work, not homework. So you need to do a little bit of rough work on it, and I'll teach you how to do that, and then of course do the paper 5. So the paper 5 is only 30 marks, and this is a very tough paper. I consider it one of the most difficult papers in A-level biology. So paper 2 and 5 on the same day, but 2 of course is the AS level, and some of you might have taken it and taken the exam. Some of you might be doing the composite, which is all the five papers together. So it depends on what you are doing and how you are going to be doing it. Either you're doing just the 2, you could be doing paper 1, 2 and 3, which is of course the AS level, so 1, 2 and 3, or you could be doing the 4 and 5 later on, which is the A2, or you could be doing all 3 of them together. Well, of course, I prefer all 3 of them together because I feel you have a better know-how and you know biology a little better then. Chlorella is a photosynthetic or single-cell protoctist. So chlorella, photosynthetic single-celled protoctist. So we know it is a eukaryote that lives in ponds. A group of students decided to investigate the effect of temperature on respiration in chlorella. So understand that they said it's a photosynthetic organism, but what are we investigating? The effect of temperature on respiration in chlorella. Now this is somehow, a student would think we're talking about photosynthesis because he didn't read the question carefully and he didn't even understand the question carefully. The students were provided with test tubes, metal foil, flat bottom tubes with lids, glass beakers, hot water supply, thermometers, timer, pH probe and a meter, hydrogen carbonate indicator solution in a closed container, a pump connected to an oxygen supply, a pump connected to a carbon dioxide supply, and a suspension. Now I always ask you this question and I say, what is the difference between a suspension and a solution? And very few of you can give me the answer to that. A suspension of chlorella with a known cell density. You know, cells will not dissolve. Solution is if you add sugar to water, sugar dissolves. If you add sodium chloride to water, sodium chloride dissolves in the water. Suspension means there will be bits of this suspended because the chlorella is a protoctist, so it has a cell membrane. Cell membrane is a phospholipid bilayer. Now that is not going to dissolve. So please understand the definition of suspension. Carbon dioxide is an acidic gas, so it causes the pH of the hydrogen carbonate indicator solution to change. The indicator solution changes color as the pH changes. So as the pH changes, the color changes. Now this is all the information that they're giving you. You need to comprehend it. You need to read it very carefully. Figure 1.1 shows the range of colors seen in hydrogen carbonate indicator exposed to different carbon dioxide concentrations. Now, this is the most important point. If we were studying photosynthesis, carbon dioxide would decrease. But we're studying respiration, so carbon dioxide has to increase. Now, let's read the question. Figure 1.1 shows the range of colors seen in hydrogen carbonate indicator exposed to different CO2 concentrations. Now atmosphere carbon dioxide is somewhere here, this point. And then there is red, and then there is magenta, and then there is purple. And on this side we have orange, and then we have yellow. So if you see now what is happening, this side is increasing carbon dioxide and this side is decreasing carbon dioxide. So please understand this. What do we mean by increasing or decreasing carbon dioxide? Now, if you were given a question on assessing photosynthesis and studying the rate of photosynthesis, well, you would see it on this side because the carbon dioxide concentration would decrease as there would be more photosynthesis. So more photosynthesis, carbon dioxide would decrease. Now, increasing carbon dioxide would mean more respiration. So this is what you have to remember, is that respiration produces carbon dioxide and photosynthesis uses carbon dioxide. So I remember there's another paper 5 in which this was used to study the effect of the rate of photosynthesis. The students made a series of color standards from yellow to purple by bubbling different volumes of carbon dioxide gas into the indicator solution of the pH solution. The pH was measured with a pH meter for each color, and the solutions were sealed in flat bottom tubes labeled with their pH. So understand what they did. They made color standards from yellow to purple. You know, you have the scheme yellow here and then you have purple here. And they bubbled volumes of carbon dioxide and the pH was measured in each color. So the student used the indicator solution to investigate the effect of temperature on carbon dioxide production during respiration in chlorella. You must realize all plants respire and photosynthesize. So photosynthesis has to be more than respiration during the day when there is a lot of light. Now it says suggest a hypothesis. Suggest a hypothesis. Now what is a hypothesis? A hypothesis is a theory. Suggest a hypothesis that the student could test in this investigation. So I mean, what can they test? We are studying the effect of temperature. So what can we test? We can test something like as temperature increases, the carbon dioxide increases or increases and then decreases or levels off. So you could have come up with any such situation in which you could have said, okay, well, I think that as the temperature is going to increase, the carbon dioxide production will also increase. Why? Because there will be more respiration, and of course there's going to be more respiration means more carbon dioxide produced. Now, part 2 of the question says state the independent variable and the dependent variable. Now, first of all, I want you to understand is what is the independent variable? So the independent variable is something like, I say, always give you an example. I say, well, you are independent. Well, I hope you are independent. So you are independent. You means what are you going to change? What are you going to change, and what different in this situation, what are you going to change? So you're going to change the temperature here. You're going to take maybe 5 different temperatures, 30, 40, 50, 60 or 70 or something like that. So what are you going to change? In the practical, when you go to the lab, what are the things that you're going to change? Dependent variable is always and always the result. What are you going to measure? Are you going to measure the bubbles of gas produced? Are you going to measure the time taken for something to change color? Now here, what are we going to look at? We're going to look at the color. So the student used the indicator solution to investigate the effect of temperature on carbon dioxide production. And then we talked about the color. So we're going to be measuring the color, and we're going to seeing the color, and we're going to see, okay, the pH or the color, that is what was allowed in this as the marking scheme for dependent and independent variable. So dependent and independent variable you need to be very clear about. Independent is what you change, and dependent is the result. What are you going to be measuring? Right? So I will try to do another video on dependent and independent variable as well, so that you can all figure this out easily. Then we come to question part 3 of this question. Describe how the students could use the apparatus provided to investigate the effect of temperature on carbon dioxide production during respiration in chlorella. Your method should be set down in logical order and be detailed enough to let another person follow it. Details of making the color standard should not be included in your method. Right? So the first thing which I tell you is that you need to imagine that you're actually doing this experiment in the lab. Now, in order to have that vivid imagination, I'm just going to do a sort of a simulation with you, and maybe you can understand what I mean by this. Now the first point is, let's take 5 different temperatures. Now that is the procedure that I always ask you to do, is that whatever you're going to change, as you're going to change the temperature, which is the independent variable, so you need to take 5 temperatures. If you were going to change the pH, take 5 pH. So we take 5 different temperatures, and you said, okay, setting up at least 5 water baths. So we have 5 water baths here, which I've set up in the lab. We're doing this experiment in the lab. Just imagine you're doing it in the lab. So 5 water baths at different temperatures, you got a mark for that. And then if you gave me all these, if you gave me these 30, 35, 40, 45, 50, and you gave me the centigrade, the unit with it, well, you got another mark. So 1 mark. So in this diagram, setting up 5 water baths and giving me the temperature of the water baths got you your 2 marks. Next is, let's take the indicator solution. Now the indicator solution we take in 5 test tubes, and I've just drawn the indicator solution, and look what I've specified. I've specified the volume. I always say whenever we're talking of a liquid, must specify the volume, 5 ml, 2 ml, 3 ml, and the concentration. So I say, okay, you could have done anything. I'm not saying you had to do this, but you have to specify a volume and the percentage. Now, saying that you would take 5 ml of the indicator solution and 1% of the indicator solution, well, you got another mark for that. Now, another thing which you would do is you would add oxygen to the indicator solution. Why would you add that? Because you see in the apparatus it had said a pump connected to an oxygen supply. Why? Because when you're going to add chlorella, where is the oxygen going to come for it to respire? So adding oxygen, and of course because it was mentioned in the apparatus, a pump connected to an oxygen supply. So adding oxygen to the indicator solution got you another mark. Next thing we needed to take was the chlorella. Now in the paper in the beginning it had said a suspension of chlorella with a known cell density. So this was given to us. And now what had we to specify? We only had to specify the volume. So you could have said 2 ml. So if you said 2 ml or 3 ml or 5 ml, well, that is what you were going to specify, that for all the 5 different temperatures, you were going to keep the volume the same, and the cell density, of course, there was only one given to you. So that was given in the paper in the beginning. Now, as you're imagining you're doing this in the lab, so now you will put all of these, you will put what? You will put the indicator and the chlorella. So the chlorella goes into each of the different temperatures. So we have 5 different temperatures, so 5 different test tubes, all with 2 ml chlorella. So they go into each of this. You can see here I've drawn this. The color coding is very important. I can keep the same color. And also you've kept the indicator. Now, putting the chlorella and the indicator in separate tubes in a water bath to reach the desired temperature, because now you are ensuring that the chlorella and the indicator and the chlorella both are at the same temperature. So you've kept them separate first, and then when they reach the desired temperature, and then of course comes the point when you're going to mix this. And when you mix this, you remember you have to then keep them in a foil, because you don't want them to be exposed to light, and you want to cover them with foil, and then you're going to wait for a certain specific end point. And that end point, you have to decide what is that end point. So we are doing this in the lab. Imagine you're doing this in the lab and think of all the points, and I hope this is going to help you in doing the paper 5. The specific end point could be the fact that how much time does it take to become yellow, which is of course the increasing carbon dioxide concentration. Then the next is, what would you use as a control? Now control means as a comparison. So control would be that, because the living organism in this is the chlorella, so what you would do is you would boil the chlorella. So boiled chlorella, and if still the same reaction was taking place, then that means there's something wrong. So we would use boiled chlorella of the same volume. That is important, is the same volume. So boiled chlorella of the same volume, which we've used here. And then of course that would be the control. Then there's something which you write nearly in every practical, and that is repeat. Repeat means what? Repeat means the results become more reliable. Repeat means that you repeat the experiment 5 times, and then you calculate the mean. And sometimes there are anomalous readings. Anomalous reading means those which don't fit a trend. Like you're getting a reading of say 10, and then 11, and then you get 12, and then you get a reading like 30. Now there's something wrong, because you see the trend is something like this, and then so you remove the anomalous reading. If you only took one reading and that was 30, well then you're probably doing the anomalous reading. So when you do a repeat experiment, that makes it more reliable, and that is what you have to understand, is repeat the experiment. At least I say 5 times. In the mark schemes it's 3 times, but I say 5 times, and to eliminate or remove anomalies. And then the last thing is that how risky is this experiment? Now this was a very low risk experiment. Why? Because you weren't handling anything very dangerous, no chemicals. But the only thing you need to write is that because you were handling plant matter, you could be allergic to something. So you use gloves when you are handling the chlorella, and that is something which you need to write. You have to write a safety precaution. That is one of the most important things which you need to give, so that you get your full marks in this designing experiment. Now I'm giving it to you in writing because maybe you've understood it, but then you don't know how to write it. Because some of you are very good, you understand it, but then your English is a little difficult to comprehend, or you are not very good at, putting your thoughts into words. I mean, we all have our problems. Sometimes I struggle with it as well. So it's okay. There's nothing wrong. We just accept our strengths and weaknesses. So number 1, 5 water baths at different temperatures, and then you said 30, 35, 40, 45, and you wrote Celsius. You got a mark for that. So 1 mark, another mark. So 1 mark here, 1 mark here. Then the volume and concentration of the indicator solution you specified. Then you're adding oxygen into the indicator. Then indicator starting at the same pH. Then mix and start timing. So you mix the chlorella and the indicator and you start timing. Then the same volume of the chlorella. I just explained that to you earlier, but I'm just giving you the wordings. Then put chlorella and indicator in separate test tubes in the water bath to reach the desired temperature, and you leave it for 5 minutes. And I always say that, like for instance, you've a Nestle bottle lying outside and you keep it in the fridge, but it's going to take some time for it to get cold. So you leave it for 5 minutes so that it becomes the desired temperature. We talk of that. Then you mix the chlorella and the indicator. Naturally, now you're going to mix that is what was given in the question. So things from the question you're going to pick up. It's not things that you've got to think of and come off from somewhere else. It's just things from the question. So you mix the chlorella and the indicator, and then of course you talk about a control, which is nearly in every practical you talk about a control. So when a control, it means a comparison. Like if I say, the enzyme is causing this reaction, so then you boil the enzyme and then do the experiment. If it still works, then it was something else, it wasn't the enzyme. So control, use the boiled chlorella, then repeat at least 5 times. In the mark schemes it says 3 times, but I say 5 times just to be on the safe side if CIE decides to change that. And why do we want to do that is, number 1, to make the results more reliable, and number 2, to eliminate anomalies. Anomalies means error, maybe a human error, maybe an experimental error, and you've got a reading and it doesn't follow the trend, and of course we talk of that. Then it's a low risk experiment, and so it's the allergy. Maybe when you're handling chlorella, you could be allergic. Like I'm allergic to mangoes, so if I just touch a mango, even I get a rash. So low risk, allergy, use gloves. And I hope now that this designing is a little better now. The B part of the question, it said here, the B part 1, state how the student would calculate the rate. And 1 over time is the best to calculate the rate. So the the time taken to reach the end point, 1 over that, and you got that rate of these. This of course comes in number of paper 5s, and they keep on asking you this. Then it says sketch a graph on figure 1.2 to show the expected result of the effect of temperature on the rate of respiration of chlorella. Now this was for 3 marks, so you have to realize 3 mark schemes were what? For temperature on the x-axis, labeled axis and units. So you labeled the axis and you got the units, well you got 2 marks. Then an appropriate curve, because as we know, temperature is going to respiration is an enzyme controlled reaction, so more the temperature to reach the optimum, and then it has to denature. So that would be a typical graph which shows that respiration is an enzyme controlled reaction. Then we come to the C part of the question, and the students carried out a second investigation to determine the effect of cell density on the carbon dioxide production of suspension of chlorella. All the suspensions were kept at a constant temperature, The student used a probe to measure the production of carbon dioxide in the suspensions of chlorella. The results are shown in table 1.1. Cell density in suspension, arbitrary units 10, 20, 30, 40, 50. Carbon dioxide concentration after 15 minutes in milligram per dm cube. After 15 minutes. Please read that very carefully. After 15 minutes. And then it's a rate of carbon dioxide production in milligram per minute. So this is now 15 minutes and this is per minute, of course, but when we read it we are not going to be careful and we're going to miss this out. And then it said you have to complete this, so this is 0.31, the reason being 4.7 divided by 15. Then part 1 of it said complete table 1.1 by calculating the rate of carbon dioxide for the cell at 40. I've just done that with you earlier on. Then we come into this part. The student used Pearson's linear correlation to test the hypothesis. As cell density increases, please underline these words, increases, the rate of carbon dioxide production increases. Now, null hypothesis means you negate it. Negate means you nullify it. You say, no, it's not correct. So there is no significant, it's very important that you write this, there is no significant correlation between cell density, which I just underlined here, and the rate of carbon dioxide production. You say there is no relationship, there is no significant relationship or no significant correlation. You could have also said that the relationship between cell density and the rate of carbon dioxide production is not significant. So cell density and the rate of carbon dioxide production is not significant. So null hypothesis means you have to negate it. You have to say this is not happening. This is not actually true. And then it says part 3, the Pearson's linear correlation coefficient was calculated at 0.25. State what the calculated value R is equal to 0.85 indicates about the results. So you read this very carefully and it says was calculated, it was 0.85, and what does this say? Now as you know it is nearing 1, 0.85 is nearing 1, so when it's nearing 1 there is a strong positive correlation, and that is what you have to revise it, that it is, you see, if it is nearing 1, what does it say? Is it nearing 0, then there's no relation. So positive correlation, and it's a strong positive correlation. Now, in this question, you had to, whenever you're asked to do this, is that calculation may not be valid. When you have to invalidate it, or you have to say that, okay, this doesn't make sense, this is not a very valid conclusion, you have to actually find faults in the experiment. You have to find faults in the data which was given to you. So now the data that was given to you was very limited. Look at this data given to you. The cell densities were only 10, 20, 30, 40, 50, and then we only had 1 values here. We didn't have a mean that we did it 5 times and then we calculated the mean. So we got these 5 values after 15 minutes. We didn't test it after 5 minutes or 10 minutes or 15 or 20, 30, and we had a lot of data from which to conclude all what we said. Now look at the points. Now all I told you was that when you have to say it's not valid, you've got to actually find faults in it. So correlation does not mean causation. If you say there's a correlation between the number of hours studying and getting a grade, I won't agree with you on that, because that is nothing. It doesn't make any sense. You might study for 10 hours, but you've only concentrated for 1 hour. So correlation does not mean causation. Then there was no repeats in this experiment, so we did not repeat any experiment. Then only a narrow range of cell densities were used. Then there might be other microorganisms present which are also respiring. There could be some bacteria present which are respiring and producing carbon dioxide. We didn't ensure that there was sterilization and there were no bacteria present. And then of course there are fewer than the number of paired observations recommended for the Pearson's test. Pearson test specifies the number of paired observations which must be present in order to conduct the Pearson test. So they were not sufficient enough. So this finishes this question. I know I've taken a lot of time on this one, but this is the first paper 5 that I'm uploading, and I hope you all can benefit from it and learn from it. The second question will be done in the second part of this, which will come in a day or two as soon as I can manage to finish it. And thank you very much. And I hope you all have enjoyed this, and I hope you all have learned something in this video. Now, sorry, I just want to continue with this, explaining to you the independent and the dependent variable. Now, another way of understanding is this is independent variable is what I change. So independent you the experimenter, what you change is you're changing the temperature in this experiment which we've just discussed, which I change in the experiment, and this is always put on the x-axis. So I've always made a mnemonic for this for your paper 3. It is always the first column in a table. So if you have a table, the first column is always the independent variable, and that is the independent is always plotted on the x-axis. So if on a graph you have anything on the x-axis, that is the independent variable. The dependent variable is always the result. So dependent, d-dependent are result, and it's always plotted on the y-axis. And what is the dependent variable is what you measure in an experiment. Are you going to measure the bubbles per minute? Are you going to measure the time taken for the color change? Are you going to measure the length of a column of gas? So what you measure, that is always the dependent variable. So if you get these 2 marks right, well, you've got an edge now, because paper 5 checks whether you've understood independent and dependent variable. So this is something which I wanted to explain to you in this first video on paper 5. Thank you very much.